ओम धन धन्वंतरे नमः। रागादिरोगान् प्रदतान् रक्तान् अशेषकायान् प्रदतान् शेषान् औत्सुक्यमोहान् किदान् जघान योगोरवैद्याय नमोस्तस्मै। So, secretory glands in GIT. Secretory glands include salivary gland, that is, parotid gland, submandibular gland, and sublingual gland, and some glands in the buccal mucosa. Next, gastric glands, intestinal glands, and liver and pancreas. First, let us discuss parotid gland. Histology. The parotid gland is a compound tubuloacinar gland composed exclusively of serous acini that produce thin, watery saliva rich in amylase. Histologically, it features darkly stained pyramidal serous cells, prominent branching striated ducts, and a high concentration of adipose cells that increase with age. The lobulated structure is enclosed in a dense connective tissue capsule. Key histological features. Secretory units, that is acini, exclusively serous, they contain pyramidal cells with basal round nuclei and apical secretory granules that stain darkly with eosin, that is eosinophilic. Duct system, highly developed, composed of intralobular striated ducts lined by cuboidal or columnar cells and intercalated ducts which merge into interlobular excretory ducts. Connective tissue. The gland is enclosed in a capsule and divided into lobules by septa, which contain blood vessels, nerves, and larger ducts. Adipose tissue. Mature human parotid glands contain a significant number of adipocytes, fat cells, distributed throughout the parenchyma. Myoepithelial cells, located between the basal lamina and the acinar cells. These contractile cells help propel saliva. Lymphatic tissue. The parotid gland is unique among salivary glands for having intragranular lymph nodes and lymphatic tissue. The parotid gland appears as a highly organized, dense and basophilic structure when looking at the basal nuclei with abundant secretory granules, presence of fat cells, and lack of mucus acini distinguish it from other major salivary glands. So, this is the histological picture of a parotid gland. You can see serous acini, no mucus acini. And the slide showing parotid gland is serous acini. Adipose tissue, ducts, intercalated ducts. Next comes submandibular gland. The submandibular gland is a mixed compound tubuloacinar gland composed of roughly 90 to 95% serous cells and 5 to 10% mucous cells, producing a protein-rich, strictly viscous saliva. It is organized into lobules separated by connective tissue septa and features prominent serous demilins, scabs on mucous cells, and characteristic striated ducts. Key histological components of a submandibular gland. Parenchyma composed of acini, secretory units, and a duct system. Serous cells, acini. Darkly stained basophilic base, eosinophilic apex due to high protein production, primarily secreting amylase. Mucous cells and acini. So here both serous and mucous glands are there. Lightly stained with a foamy or clear appearance. Nuclei are flattened and basally located. Serous demilins. Mixed acini often appear as serous cells, forming a cap moon shape over mucous cells, which are considered a classic histological artifact of preparation. Duct system contains long intercalated ducts, prominent striated ducts, intralobular, and interlobular excretory ducts. Myoepithelial cells, contractile cells located between the basal lamina and acinar cells, aiding in saliva secretion. Functional organization. The gland is divided into lobules by connective tissue. It acts as a mixed gland, serous and mucous, with serous cells predominating. Intercalated and striated ducts help modify the inorganic components of saliva, that is electrolytes. Submandibular gland. Coming to these are other slides of submandibular gland. You can see both serous and mucous secreting acini. So both serous and mucous secreting gland. Next is again next is sublingual salivary gland. This is a pure highly, it is a mucus secreting gland, predominantly mucus secreting gland. You can see it. Sublingual salivary gland is a mixed gland, predominantly mucus, that is 65%, and minor serous components characterized by light staining, foamy mucus secreting acini. It is a compound tubuloacinar gland containing prominent mucous tubules capped by serous demilins. The ductal system is less complex than other major glands with short intercalated ducts and rare or absent striated ducts. Key histological features of sublingual gland. Acini structure, primarily composed of mucous cells which appear as pale foamy cytoplasm with flattened nuclei pushed to the base. Serous demilins. Darkly stained serous cells form caps, demilins, on the ends of the pale mucous tubules. Ductal system of the sublingual gland contains intralobular ducts, intercalated and some straight within the lobules, and interlobular ducts within connective tissue septa. Connective tissue. Gland is surrounded by a thin connective tissue capsule and divided into lobules by septa. Differences. It is easily distinguished from the submandibular gland because it is mainly mucous, not mixed serous, and lacks numerous well-developed striated ducts. Structure and function. Sublingual glands are located on the floor of the mouth and produce a thick, mucinous saliva that provides lubrication. They are considered the smallest of the major salivary glands and are the only ones without a well-defined caps. Some more slides of sublingual salivary gland. Next, coming to liver histology of liver. Liver histology examines the microscopic structure of the liver, which is organized into lobules composed of hepatocytes, sinusoids, and portal tracts. Key components include hepatocyte plates radiating from a central vein and portal tracts, this bile duct, portal vein, and hepatic artery at the periphery. Specialized cells like Kupffer cells as macrophages and I2O cells, stellate cells, are also crucial components. Key histological structures of liver. Hepatocytes, large polygonal parenchymal cells with eosinophilic cytoplasm and prominent nuclei. Portal tract, located at the corners of the classic hexagonal lobule, containing a portal venule, large lumen, hepatic arteriole, thick wall, and bile duct, simple cuboidal epithelium. Sinusoids, specialized fenestrated capillaries between hepatocytes that transport blood from the portal vein and hepatic artery toward the central vein. Spaces of Dissay. Perisinusoidal space located between hepatocytes and sinusoids where nutrient toxin exchange occurs. Bile canaliculi, tiny channels between hepatocytes that transport bile toward the bile ducts. Structural organization units. Classic hepatic lobule, hexagonal shape with a central vein in the middle and portal tracts at the corners. Portal lobule, triangular based on bile flow with a central portal tract and central veins at the periphery. Hepatic SNS, diamond-shaped unit based on blood perfusion or oxygenation consisting of three zones. Zone 1, periportal, gets the most oxygen, while zone 3, centrolobular, gets the least, making it more susceptible to ischemic injury. Common stains in liver pathology, hematoxylin and eosin. Routine stain, revealing general architecture and hepatocellular details. Mason's trichome highlights collagen stained blue to stage fibrosis, scarring, which is vital for chronic liver disease assessment. Reticulin stain outlines the reticulin fibers, stained black, to analyze the liver plate architecture, crucial for detecting liver cell plate disruption. Next is Pell's iron stain, used to identify iron overload, same as hydrogen. Liver histology is essential for diagnosing, staging, and determining the prognosis of various diseases, including cirrhosis and hepatitis. Some more slides of liver histology. Next is pancreas histology. The pancreas is a mixed exocrine endocrine gland with its lobulated structure. Over 95% of its volume is composed of serous acinar cells, exocrine, arranged in tubulo-alveolar units that produce digestive enzymes, while 1 to 2% comprises islets of Langerhans endocrine scattered throughout producing hormones, insulin, glucagon, somatostatin. Exocrine pancreas, Acinian ducts. Acinar cells, pyramidal shaped serous cells arranged in acini, characterized by basophilia, rough, and apical zymogen granules, precursor enzymes. Duct system. Enzymes drain into intercalated ducts, which merge into intralobular and then interlobular ducts. Centroacinar cells. Pale staining cells within the acinar lumen, unique to the pancreas, representing the start of the intercalated duct. Duct cells. Secretory, cuboidal or columnar cells that release bicarbonate and water to neutralize stomach acid. Endocrine pancreas, islet of Langerhans. Islet of Langerhans, pale staining, spherical clusters of endocrine cells scattered among the acini. Cell types contain alpha cells, beta cells, insulin, most numerous, delta cells, somatostatin, and PP cells. Vascularization. Highly vascularized with fenestrated capillaries for hormone release directly into the blood. Stroma, thin connective tissue capsule. Septa, separating lobules containing blood vessels, nerves, and ducts. So some more slides of pancreas. Say both endocrine and exocrine gland. Major exocrine, secreting hormones. You can see acini, ductules, and islet of Langerhans. Clearly you can see the acini, serous acini, ducts, and fibrous tissue, islets of Langerhans. You can see in the center there is islet of Langerhans. You can see in the center that is islet of Langerhans, containing alpha, beta, and cells. Next, coming to stomach. Stomach contains also glands, chief cells secreting pepsinogen, and oxynic cells secreting hydrochloric acid. The stomach wall consists of 4 main layers: mucosa, submucosa, muscularis externa, and serosa, specialized for secretion and mechanical digestion. Mucosa features gastric pits or fovulate, leading to glands that secrete acid, enzymes, and mucus. The muscularis externa is unique for having 3 smooth muscle layers for churning. Physiological layers of the stomach. First one is mucosa, innermost layer with simple columnar epithelium that dips into gastric pits and connects to gastric glands. It includes the surface mucous cells, lamina propria, and muscularis mucosae. Second one is submucosa, contains connective tissue, blood vessels, lymphatic vessels, and nerves, submucosal plexus. Third one is muscularis externum, composed of 3 layers of smooth muscle, inner oblique, middle circular, outer longitudinal, to facilitate complex journey. Fourth one is serosa, the outermost layer composed of simple squamous epithelium, mesothelium, and connective tissue. Gastric glands and cells. The gastric mucosa changes by region, cardia, fundus or body, and pylorus. Fundic glands are primarily found in the body and fundus. Parietal or oxyntic cells, large pink staining cells that secrete hydrochloric acid, an intrinsic factor. Chief or peptic cells, third one, dark purple basophilic cells that secrete pepsinogen, which breaks down proteins. Mucous neck cells, found in the neck of the gland. They secrete a different type of mucus than surface cells. Enteroendocrine cells, for example, G cells, secrete gastrin, and ECL cells secrete histamine. Regional variations. Cardia primarily contains mucus secreting glands. Fundus and body contains gastric glands rich in parietal and chief cells. Pylorus pits are deeper. Glands are primarily mucus secreting, protecting the small intestine. Some more slides of stomach histology. You can see stomach glands containing peptic chief cells, and synthetic cells, and mucus secreting cells. Region-wise, it is structured. Body, fundus, and pylorus. You can see gastric pits. Next, small intestine glands. Intestinal glands, crypts of Lieberkühn, are simple tubular invaginations of the mucosa between villi, small intestine, or surface epithelium, large intestine, extended down to the muscularis mucosae. Lined with simple columnar epithelium, they function in secretion, mucus, hormones, and enzymes, and epithelial renewal. Key cellular components, enterocytes, absorptive cells, most common. Goblet cells secrete mucus, increasing in number toward the distal intestine. Then Paneth cells, eosinophilic granular cells at the base, secrete antimicrobial agents like lysozyme and the defensives. Enteroendocrine cells secrete hormones like secretin, CCK, and gastrin into the bloodstream. Next, stem cells located at the base responsible for rapid epithelium regeneration. Regional variations. In small intestine, crypts exist alongside villi. Duodenum has Brunner's glands, submucosal. Jejunum and ileum has Peyer's patches, lymphatic. Large intestine. In large intestine, deeper, parallel, test tube-like crypts, more goblet cells, and no Paneth cells, usually. Goblet cells secrete mucus. These structures constitute a dynamic factory where cells differentiate at the base and migrate upwards toward the lumen in a few days. Some more slides of small intestine. You can see in the small intestine, between the villi, there is invagination of mucosa containing glands. You can see villi and crypts of lymph. In the crypts of lymph, some secretory cells are secreting enzymes. So they have discussed different glands, secretory glands in the gastrointestinal tract. So thank you for patient.